So today I want to talk about a project we've been working on the isolation and immortalization of PRRS GP5 specific porcine B cells. As a little bit of background of humoral immunity against PRRS, we know that PRRS specific antibodies can provide a sterilizing immunity, but we know that the development and quality of the neutralizing antibodies varies widely from pig to pig. But we do know that cross-protection and broad neutralization are possible under field conditions, albeit they are very rare. So one of the main questions we're asking in our lab is, how is PRRS virus neutralized? Is it a polyclonal or a monoclonal antibody response? What viral epitopes are involved? How are broadly neutralized antibodies generated? And as a little side thing, are non-neutralizing antibodies, do they play a role in the reduction of viremia? So to test, or to look into some of these questions, we designed an experiment where we take PBMCs from pigs that show evidence of broad neutralization against PRRS. We isolate IgG-positive memory B cells. We immortalize these B cells and create PRRS-specific cell lines, and then we characterize their secreted PRRS-specific antibodies. So first, we selected 2 pigs. I'll refer to them as BNW4 and BNW7 for the rest of the talk. Both of these pigs are parity 1 sows. Each pig has seen multiple ATP vaccinations and either challenged with 1 or 2 PRRS2 field strains. But after all of this, both of these pigs show broad neutralization against all PRRS2 and PRRS1, so they are hyperimmune to all PRRS virus. And even 90 days post the last virus exposure, they retain really high level neutralizing titers. You can see over here in blue we have BNW4, in red we have BNW7. On the far right is the neutralization titer against ATP, which is homologous virus. They're vaccinated against this. And we're about at 1 to 8,000 neutralizing titer. Next to that, the next blue line is neutralization of VR232, and technically that's heterologous virus, these guys, because they never saw MLV. So they're about 1 to 4,000, so very high neutralizing titers. For BNW7, for ATP, we have about 1 in 2,000. For VR232, about 1 in 164, so very high neutralizing titers for both these pigs. So we extracted PBMCs from these pigs. We sorted them based on flow cytometry, based on CD21 and IgG. After we sorted them, we immortalized them using proprietary retroviral vector developed by AIM Therapeutics. And the nice thing about this vector is it allows the B cells to retain their B cell receptor and at the same time secrete large amounts of high concentration antibody. And then also they express GFP, which makes them really easy to resort after they've been immortalized. And that's exactly what we did next. We resorted them based on their GFP expression in IgG into 20 B cell populations, and then we screened these populations for PRRS specificity based on IFA. And so the IFA we designed, we take marked cells, we infect them with virus, we fix them, and we throw on supernatants, which contain antibody from all these different B cell populations, and we screen for PRRS virus specificity. And if our B cells secrete antibodies that are PRRS specific, we get these nice little niduses of infection that light up. If they are non-specific, you get nothing. So through that process, we identified 5 PRRS specific B cell lines, 1 of them from BNW4 and 4 of them from BNW7. And it should be noted, I do not believe these are the whole B cell repertoire of these pigs. It's probably just a little tiny pie slice of them, but we did find some really interesting results with these antibodies. So now that we have these PRRS-specific B cell lines, our next objective is to evaluate their secreted antibodies. And to do this, our first test we looked into was to see their binding capacity to different PRRS viruses. And we did that through a similar IFA assay to what I just showed you, where we infected marked cells with a bunch of different PRRS virus here, and we saw basically where does the antibody stick. And just as a reminder, these pigs they saw ATP, and also both pigs saw a 134 PRRS2 virus. And then what we found out was that our isolated antibodies were actually broadly binding to all PRRS2 virus. On the left here we have serum, and then we have all of our different isolated antibodies. On the top we have a variety of PRRS1 and PRRS2 viruses. So every isolated antibody we had was broadly binding. And despite also, I mean, we got to remember that both of these pigs up here, they're broadly neutralizing against all PRRS virus. So they can bind every single antibody with their serum. So next thing we looked into was protein specificity, viral protein specificity of these isolated antibodies. And surprisingly, we found that 4 out of 5 of them were GP5 specific. One of the antibodies, we could not identify what protein is specific to, but we believe it's a non-structural protein based on the IFA staining. And one other thing to note is we're missing a lot of proteins over here. This is just what we had available in the lab, recombinant proteins. So there are other proteins, especially like GP2, 3, and 4, that are of high interest that are missing here. So the next thing we looked into was neutralizing capacity of a select few of these antibodies. And so I took P5C4, which was a GP5 specific antibody, and then P10C5, which was the one we could not identify what viral protein it was. And we took these monoclonal antibodies, we tested the neutralization against homologous virus. And what we found out is the GP5 specific antibody, P5C4, could neutralize virus about a 1 to 8 dilution. But the other antibody, P10C5, was incapable of neutralizing virus at the same antibody concentration. However, when we went over to heterologous virus, VR232, neither antibody could neutralize the virus, which was really interesting. Despite that P5C4 being broadly binding, it was only homologously neutralizing, which was fairly surprising. Another thing we looked into was how the VDJ genes of the heavy chain for P5C4. What we saw was there was a high level of somatic hypermutation in these heavy chains, about 13 to 20% divergence from historical variable region genes, which isn't too surprising. Past research has shown there's high levels of somatic hypermutation in pigs in response to pathogen, but nevertheless, it's really interesting, and I think it actually it's a pretty big piece of neutralization of PRRS. So in conclusion, we saw a few things. We saw that all isolated antibodies bound every PRRS2 virus tested, but not PRRS1, and this was seen in both neutralizing and non-neutralizing antibodies. And then also our defined PRRS-specific antibody populations were dominated by these GP5 specific clones. We also saw that isolated GP5 specific monoclonal antibody P5C4 was able to neutralize homologous but not heterologous PRRS virus, despite it being able to bind all PRRS2 virus. Then we also saw that heavy chain variable region sequences showed high evidence of hypersomatic mutation. So what does this mean about PRRS neutralization? Is it a polyclonal or is it a monoclonal antibody response? All of our isolated antibodies were only homologously neutralizing. And to us this suggests, especially since the serum could also neutralize PRRS1, is that this is the response to virus is polyclonal. So there's a lot of different antibodies at play neutralizing PRRS. What viral epitopes are involved? We saw that GP5 specific clones, they dominated our isolated antibodies. But this doesn't necessarily mean that they're the only viral epitopes that are involved in neutralization. And we're looking into those other epitopes right now, looking at 2, 3, and 4 also. And then also we were looking at how are broadly neutralizing antibodies generated. And one thing we saw was that we saw these broadly binding antibodies, but that weren't necessarily broadly neutralizing. And we saw these high levels of somatic hypermutation. So we suggest that maybe there's a combination of both those together. This capacity to produce broad binding and highly mutated could eventually lead to broadly neutralizing antibodies within the pig. And I think they do exist within the pigs we did test, but we just hadn't isolated them. And then also as a little side, do non-neutralizing antibodies play a role in the reduction of viremia? And we saw this interesting dynamic in our antibodies, that they were broadly binding, but not broadly neutralizing. And depending on which virus the pig was exposed to, an antibody that was homologously neutralizing or neutralizing could become non-neutralizing when you switch viruses. So to us, this suggests that maybe non-neutralizing antibodies actually play a bigger role in the production of viremia than we think, maybe through secondary antibody effector functions. So to finish up, I'd like to thank our funding source, USDA NEFA grant, also the Sam Maheshwan Graduate Fellowship, which funded me as a grad student the last few years. I'd also like to thank everybody in the Murtaugh lab, especially Mike Murtaugh. He was the inspiration for this whole project. I'd also like to thank everybody at the Purebright Institute. They sort of got the ball rolling on this project, helped us get everything started. And of course, I'd like to thank AIM Therapeutics for providing the vector that made this entire experiment possible. All right, now I'll take any questions.
Question for Jordan.
Did you do any neutralization by pooling those monoclonal antibodies?
Excuse me, what was that?
Did you do any neutralization assays after pooling those individual monoclonal antibodies?
Putting them together? Yeah, I did actually. I did release that non-neutralizing clone and that neutralizing clone. I did mix them together, and I saw about the same level as neutralization. But I see where you're going, because that was one of the things I wanted to do, is start pooling all these monoclonals together and see if I get enhanced neutralization. And I do believe if I do get more monoclonals, the more I add together, I'm going to get higher levels of neutralization. That's probably beyond what you would expect of the individual antibodies. Is that what you're... Yeah, and broader, exactly, much broader too. And that's what I would expect, though, if we get more of these monoclonals.
But is it possible that the 4 that are GP5 specific are actually recognizing the same epitope?
Oh, absolutely, yeah.
And the dominant, so adding them together wouldn't change things.
And I think they are, actually. I think they're all probably looking at a conserved region, which is totally possible, probably epitope B. But the interesting thing about them is they're not broadly neutralizing. So there are other players, there are other antibodies that are involved in this neutralization process.
When you collect the PBMC, do you have, at that time point, do you have any evidence for broadly neutralizing antibody in that pigs?
Yep. Yeah. So we did retest the serum from those pigs.
How many strains did you test?
Let's see, about 8 PRRS 2s and then 2 PRRS 1s. So in all of them, they neutralized at least at 1 to 8 at lowest. And those were the PRRS 1s, yeah.
Yeah, one more question. I think Dr. Roney mentioned about the immunodominant antigen, like GP5. When you immunize them, you may need to knock out that immunodominant epitopes by using very specific non-dominant rare epitopes. Yeah. That kind of situation happened in HIV infection.
Yeah, no, I totally agree, because I think there are. I don't think GP5 is the actual neutralizing epitope. I think 2, 3, and 4 are really bigger players.
I may miss during your presentation, but do you use the antigen specific to fix the B cell, to select for the B cell, or you just clone every B cell?
Just everything. We tried to use virus specificity. We used labeled virus initially, and we didn't really get any better results than through just random selection. But we are looking into, we have a lot of his-tagged proteins, especially 2, 3, 4, and 5, and that's one of our next steps is to do antigen-specific sorting. Yeah.